Okay. Um, I have a shortened version of the learning objectives, okay? And what we're really going to be spending our time on is things that are made from amino acids, okay? Creatinine, creatine, creatinine, GABA we talked about already, serotonin, melatonin, nicotinamide, we've spoken about. What's the source? What amino acid can also give us nicotinamide? What amino acid? What? Tryptophan, very good, okay. So that we'll review it again. Then we have the whole group of neurotransmitters, dopamine, norepinephrine, epinephrine, and then these are pigments, the melanins, okay, all right. And then we have histamine, and histamine is also a neurotransmitter and also is involved in inflammation, and we need to know it. And this one is very interesting, nitric oxide, okay. What's interesting about nitric oxide, it's a neurotransmitter, okay, but it's a gas, okay? So I think one of the things we should ask you is that what is nitrous oxide used for? Not nitric oxide. Nitrous oxide. It's laughing gas, yes, very good, okay? And so what is it used for? Yeah, it's an analgesic. And it's not a very good, it's such a poor anesthetic that you would have to have 100% of what you ingested into your lungs would have to be nitrous oxide, and then with no oxygen, and then you die of nitrous oxide toxicity, okay? So, so you have to have oxygen in there, so it's an analgesic, okay, and it's used that way, okay? I haven't had a kosher dollar in the others, okay? So nitric oxide is NO. What first lady is known for using the word NO as her motto? And it's not Melania. It's not Melania to Donald. Okay. What first lady had a policy? In fact, she had shirts on it. It said, "Just say no." Nope. No. Nope. You got to go back a little further than you don't have to go back. Yes. Nancy Reagan. Very good. Okay. Okay. So NO is nitric oxide, and nitrous oxide is NO2. Okay, and don't confuse the two of them because it can be really crazy. And I know I have I wouldn't call it a one. Thank you. I don't bend over very well, so you can pass it on to him. He'll come down for it. Okay. So we're going to talk about that. Okay. So, all right. So creatine. Okay. People who go and buy things in GNC. Okay. Will know that they're doing creatine phosphate. Do you have any idea what its function is in tissue? Tissue that's muscle, for instance. What? What does it do? Somebody shakes their head, but they're not going to tell me the answer. What does creatine phosphate do? It's a storage form of energy in muscle. Okay, creatine phosphate. Okay, so it's an important molecule. So it's a storage form of that. Okay. And so it's actually going to be, we're going to make it synthesized, okay? Now, here's the molecule of creatine. Could you tell me the 3 amino acids possibly from looking at the structure? This group is a guanido group. So what do you think is going to be the donor? The only amino acid that has a guanido group, which I just told you. No. So it starts with an A and has an ene in it. Arginine. Janine, okay? All right. And where's the methyl group going to come from? 99.90 SAM, right? And that comes from what amino acid? Methionine. Okay, and then what's left? NCH2COOH. What is that? A 2-carbon amino acid. There's only one. It's glycine, right? So it's going to be made from those 3 amino acids, okay? Glycine, right? Why don't you see that? You're going to wrinkle your brow.
Oh, no.
Okay.
See that in the structure.
Okay. Okay, you see it here? You don't see glycine in the whole chart? Glycine is C2.
Yeah.
Yeah, okay. All right, here we go. So we're going to start with glycine, okay, and this is in the kidney, and we're going to add arginine, and arginine, when it donates its guanidino group, as it does in the urea cycle, what's left, the other piece is ornithine, okay? So we're going to get that, and we're going to have this, and then Sam comes in, and Sam goes to SA and puts in the methyl group, and we make creatine by that method, okay, from those 3 amino acids, okay? So if you see a molecule and you can identify it, this one's relatively easy because the arginine is there. You could know that that's the way it is. Creatine phosphate is then made by creatine phosphokinase, sometimes called creatine kinase. And one of the reasons that I'm emphasizing this is that CK and or CPK, depending on just the terminology, is a measure of muscle wasting. Because this enzyme is primarily in muscle, it's also in brain. What's happening is that if you had muscle wasting, the level of CK in blood goes up, or CPK, the same one, okay? And so it's a measure of muscle breakdown, okay? Now you say, oh, that doesn't happen. It does happen, okay? And one of the issues is people who are on statins, okay? Does anybody remember what the statins were? Cholesterol, the internal cholesterol, okay? And I think I talked to you about what some of the major side effects were of the cholesterol inhibitors, the statins. I think I talked about it. Anybody know? One is actually muscle breakdown, and CK goes up. And then it can be relatively minor, okay, but really serious when it goes like tenfold increase in CK or CPK. So you're going to be looking at muscle wasting in people when you're a physician, okay? So CK is going to be measured. That's a commonly looked at enzyme, okay? So it's in blood, so it gives you creatine phosphate, okay? The other thing is that the breakdown product is non-enzymatic, it cyclizes, and you get creatinine. And you're going to look at creatinine levels, okay? Creatinine levels are going to be in patients that are, when you're looking at creatinine levels to see, which is you're going to see what the creatinine levels, and you're going to see something to do with the breakdown. So you're going to be measuring creatinine, you're going to be measuring CK. So it's not the product that you're making as much as is important. There was probably 4 or 5 years ago, people were selling creatine phosphate. Supposedly, if you were a big weightlifter and stuff, they felt that you got recuperation if you added more creatine phosphate. I'm not sure even how well it's probably absorbed on that basis. So that's why I wanted to spend a little bit of time on creatine. I think it's in 5. I think it's the 47th chapter has something about creatine. Okay? All right, next thing. Okay. So here's GABA. GABA is gamma aminobutyric acid. How many carbons? 5. Gamma amino glutamic acid. It's a derivative of glutaric acid, okay? Just like glutamine, glutamate is a derivative of glutaric acid, okay? It's got that, okay? All right? So what we're going to do is effectively, these are ways that we can make glutamate, so we can actually not deal with that, okay? But it's interesting. Glutamate is the precursor of GABA. Glutamate is what kind of a neurotransmitter? Is it 4? Oh, I'm sorry, you're correct. I was looking alpha-ketoglutarate. You're correct. It's a butyric acid, that's 4. Okay? So do you know the monocarboxylic acids? Formic, acetic, propionic, okay? Butyric, okay? So those are the 4 carbon ones. So for somebody who wants to, do I know, you want me to repeat that? So you have the 2 carbon ones. These are the single carbon ones, okay? So for instance, oxaloacetate is 2 carbons from oxalate and 2 carbons from acetate. Total number is 4. And aspartic acid is made from a 4-carbon keto acid, okay? And it goes, when you break down aspartic acid, it goes to a 4-carbon keto acid, which is oxaloacetate, okay? All right. Thank you for correcting me on that, but you should have said it out loud, okay? That's okay. No, it is 4, okay? But it comes from this, which is 5, okay? All right. So this enzyme has worked, this process as glutamate goes to this, and we're removing a CO2, so what kind of an enzyme is that going to be? Removing a CO2. Removing a CO2. It isn't a carboxylase, it's a decarboxylase. Thank you very much. I appreciate it. Decarboxylate. Okay. Now, this little thing is called the GABA shunt, and glial cells do not have this, okay? But what happens in this situation is GABA goes to effectively, what happens is here we have effectively, we've removed the, we've transaminated the amino group, and so we have this is succinyl acid, succinyl semialdehyde. Then we oxidize that to succinate, goes into the TCA. Acetyl-CoA comes in. We end up making citrate, iconatate, and then effectively isocitrate, which loses a CO2, and we get alpha Kg, and effectively we end up having this shunt. Not terribly important, but somebody in your brain chemistry course or whatever may talk about that shunt. Okay, all right. What's interesting is glutamate goes to GABA. Glutamate is excitatory, GABA is inhibitory. Okay, so we can deal with that. Okay. Okay. Now we have some of the interesting ones. Okay, this is serotonin, melatonin, and nicotinamide. Okay. Here we need to know what the starting material. Serotonin is important. What do you think, from the name, what do you think it would do to blood vessels? Sero. Anything that has sero in it means it's serum or it's blood. Yeah, it would constrict them, okay? When you tone something, you're making it, right? You're constricting it, okay? So serotonin is a neurotransmitter, but if it was working pretty much that way. And melatonin, we're going to talk about, and nicotinamide, we've already talked about. So we're going to start with tryptophan. Tryptophan is a essential or non-essential amino acid? Essential amino acid, right? Very good, okay? And it's going to be worked on by a mixed function oxidase. It's using molecular oxygen and producing water. And it has, it's a tryptophan hydroxylase, and it's using BH4. Where did we see that before? When we went from phenylalanine to tyrosine. Very good, okay, so we're going to see it. So, you know, it's the same kind of thing that we see, but it's a different reducing agent, okay? And so do you remember how does what happens to it, to the BH2? It goes back to BH4 using a BH2 reductase, okay, which uses NADPH, okay? All right. So in this situation, what we're going to do is we're going to get hydroxytryptophan. So what happens is one of the oxygens from one of the atoms of oxygen from the oxygen is going to go into the molecule, and one is going to go into water. So it's a mixed function oxidase. And we get 5-hydroxytryptophan, okay? And now what we're going to do is many of the neurotransmitters are generated by actually decarboxylating them. So we're going to work on a DOPA. This is called DOPA decarboxylase. That's from the German decision, okay? It's really the English speaking is 5-hydroxytryptophan. So it's decarboxylating this, okay? And effectively we're going to get this group coming off, and then we get serotonin, which is, has an amino group here and a hydroxyl group here, okay? And that serotonergic, there are serotonergic pathways in the GI tract. There are serotonergic pathways in the CNS. Okay, so it's a neurotransmitter. Generally tends to work in the direction of in essence, stimulating and causing contraction or on that basis. Okay, all right. It's broken down by what is called MAO-A. Do you have seen what MAO stands for? What? Monoamine. MAO, monoamine oxidase, okay? And these are going to be used again and again, okay? So, and these are the products that are going to be excreted: 5-hydroxyindoacetaldehyde and acetic acid, okay? Don't really need to know, you just know it's broken down by MAO. And then the serotonin is then acetylated, okay, on the amino group, and eventually it's using acetyl-CoA, and we get N-acetyl serotonin, okay? So here's the the next thing, we have SAM coming in, and what does SAM always deliver? Methyl groups. So this is going to be methyl and acetyl serotonin, which is called melatonin. Okay. What does if you watch commercials on TV, especially for sleeping pills and for where they're adding melatonin to them? Okay. So what does melatonin do for you? What? What? What does it do? What?
Oh, I said the wrong answer.
What did you say?
I said skin color. That's melanin.
That's melanin. That's one of the. Thank you for telling me that, because when we talk about melanin, people will confuse melanin and melatonin. Okay, so melatonin does what? Okay, so I'll tell you. Ever had the urge after a Thanksgiving dinner, okay, to take a nap, okay, all right? And the reason for it is turkey has a lot of tryptophan in it, and that tryptophan gets converted to serotonin and then to melatonin, okay? And melatonin makes you sleepy. So now what they're doing is they're actually, for people who have trouble going to sleep, not staying asleep, they add melatonin because it controls that. It also controls circadian rhythms and doing all of those things, okay? So they're actually using melatonin for controlling sleep, okay? So that's why you get sleepy after eating a lot of turkey. Maybe if you don't eat a lot of turkey at Thanksgiving, maybe it would be that way, okay? It couldn't be the wine or the grapes or anything like that, okay, that was doing. So that's an important product. And thank you for your saying melanin, okay? Because that's the one. You can guarantee that if the answer is melanin, the melatonin will be in the choice. Will be one of the choices. Okay, so good. Okay, all right. So here we have. These are neurotransmitters that are actually, and here's the melanins. Okay, so we'll come back to the melanins on that basis. Okay, all right. So here we're going to start with phenylalanine. We have to use phenylalanine hydroxylase. So somebody who has PKU deficiency, okay, is not going to be able to carry this reaction out, and to make L-tyrosine, which is why tyrosine becomes, in PKU patients, it becomes an essential amino acid. So what's the recommendation when you have an essential amino acid now? So with PKU, how would you treat PKU? How would you treat it? Restrict the phenylalanine. Get rid of it all?
No.
Why not? Right, exactly. The same way with maple syrup urine disease. You would decrease the amount of the branched chain amino acids, okay, which is really hard to do because they're a big part of the protein and stuff like that. All right? So that's an important principle, that when the choice is, you always decrease it, but you have to leave enough for protein synthesis, okay? Right? The basic principle is the tRNAs all have to be loaded for protein synthesis to occur. We're loaded with amino acids. important principle, which you didn't necessarily something that I taught you, but maybe you were taught when you were doing translation, okay? But it's an important principle that affects what's going on. Okay? So now we're going to go to this is to DOPA by sorry, I'm having a problem. Tyrosine, focus isn't great. Tyrosine phenyl hydroxylase, thank you. And you're actually putting a hydroxyl group here, and we have this, and this is called DOPA. Did anybody have heard this as a drug used for what disease? Parkinson's disease. L-DOPA is used for Parkinson's disease, okay? So it's a neurotransmitter, okay? But it primarily is important because it doesn't have a neurotransmitter. One of the reasons that it is that Parkinson's disease involves what neurotransmitter? Does anybody know? A deficiency of one of the neurotransmitters. It's the deficiency of dopamine. So you give L-DOPA because dopamine will cross the blood-brain barrier. So you effectively give them the precursor of dopamine, okay? You're giving them L-DOPA, okay? On that basis, okay? Is that making sense to people? Okay. All right. So you get dopamine, which is a neurotransmitter, okay? And notice that then dopamine is then worked on by dopamine. I have it here that I can read better. Sorry. Dopamine beta hydroxylase, okay? And effectively you get a hydroxyl group in this position, okay? And that becomes norepinephrine, okay? Notice this enzyme, okay, requires copper, okay? When we go up and look at this, okay, this enzyme, tyrosine hydroxylase, okay, requires copper in the same way. So we now have a culture or science dollar. Can you tell me 2 other proteins that use copper? Copper, like Cu, no, yeah, copper. We have to remember we have to do minerals and ions, copper, and so trace amounts. Okay, yeah. Okay.
So you want 1 or 2 of them?
I want 2 of them. I'll give you $1 for one and another $1 for the second one.
So you have cytochrome oxidase.
Very good, excellent.
And you have lysyl oxidase or lysyl hydroxylase? Lysyl hydroxylase.
It's not the one I was thinking about. I didn't say it was an enzyme necessarily. I said it was a protein. Anybody? Yes. Nitrous oxide? That makes me laughing. That's laughing gas. Nitrous oxide. No.
Oh, it's not an enzyme.
Nitrous oxide is a gas. It's not a protein. Anybody? You were absolutely right with the first
You
one.
said it's not an enzyme, though. No.
It's not an enzyme. No, it's fine. Okay. Has anybody ever heard of ceruloplasm? Did you say that?
No.
Why did you point to him? Do you think he's going to share what he makes money on?
He said he knows the answer. I never said this.
What? You didn't know the...
Totally class.
What did you say?
I didn't say anything.
Don't be shy. He wants the half of a cup of coffee he's going to get out of the $1. So, ceruloplasm, okay? What color do you think ceruloplasm is? Blue. Yes, exactly. So, all right. So who said that? Very good. You get $1 for that, okay? So at least you knew what color it was, ceruloplasm. What do you know what ceruloplasm does? There are organisms that actually have that as a nitrogen carrier. I mean, an oxygen carrier, so ceruloplasm, okay? So I didn't, those of you, do you remember that there was molybdenum? We went through that, whether it was Mo or Meeb, okay? Okay. The enzyme that uses molybdenum is actually xanthine oxidase, okay? So I don't think I emphasized that. So this is the way to look at the traces, okay? But the important one is the copper on the cytochrome oxidase. Okay, so okay, so all right. So then we get norepinephrine, which is a neurotransmitter, and epinephrine, which is a hormone, but it also can have neurotransmitter properties. Okay, so these are all made by pretty much the same pathway. Okay, and now we have this one. Okay, and remember that there were two forms of tyrosinemia. Okay, so you could have tyrosine accumulating with this one. One was called 1 and one was called 2. You don't need to know the difference, okay? And this is on the pathway to making melanins, okay? And melanin are pigments. So melanins are pigments, okay? Melatonin is essentially control over your timing, your sleep, and your circadian rhythms, okay? Everybody see that? Okay, good. Any questions about that? Okay. These occur in the adrenal medulla, okay? And some of it these occur in the, this one is in the epinephrine. You have to do it with this way, and that's the adrenal medulla. And then the neurons are effectively these pathways, okay? All right. Here's histamine. Okay. We most think about histamines as, because we take antihistamines, as we're taking, in essence, things that block the action or block their synthesis of histamine. Okay? Histamine comes from histidine by a decarboxylation reaction. Do you notice decarboxylation, making neurotransmitters, is a way to go? Okay? And then sometimes hydroxylation as well. But decarboxylate, so histidine, when you decarboxylate it, remove this, comes off as CO2, you get histamine. And this is going to occur in both brain and, we'll see in this way in brain, peripheral tissues. Diamine oxidase is a strange enzyme. It functions to, in essence, metabolize and oxidize things that have two amino groups. Do you see the two amino groups here? Here, and this one, because this it still can take a proton here. So there are two amino groups. What it really works on in the body are compounds called cadaverine and what is the other one? What do you think cadaverine smells like? Dead bodies, right, okay. Okay, it smells like dead bodies, right. Cadaverine, and there's another one, and I'm blanking on what it is, okay? But it's putrescine, okay? Both sounding disgusting words. They use diamine oxidase, okay? So if you see diamine oxidase, or if they're asking you a question about MAO oxidase or any of those things, okay? If putrescine and cadaverine aren't mentioned, then it's okay, all right? Okay, so what happens is that in peripheral tissues, the breakdown products are imidazole acetaldehyde, which then gets oxidized to the acetic acid. In brain, now, histamine, there are histaminergic neurons, okay? So there are two major places that histamine is synthesized. What would they be? In the brain is one. What's the other? What kind of cells are in, what do you think of when you think about histamine? Why are you taking antihistamines? For allergies, right? Okay. And suppose you went and ate some fish that was high in histamine. Well, that's not a good example, but you could, if I injected histamine into your skin, what would happen? Yeah. What? It gets swollen, right, because it's a vasodilator, okay? And it becomes red, and you get what's called a wheal and flare, okay? So and what do we, in common parlance, talk about those bumps that you get from histamine in allergic, say, food allergies? Hives, okay? Those are hives, okay? So it's in the peripheral tissues, and where is that coming from? What kind of cells? It comes from mast cells. Okay, you've heard of mast cells? There's a lot of histamine in mast cells, okay? And if all the histamine in mast cells was released at the same time, okay, you would end up looking like one big hive, okay? It was just a pimple with, big swelling, okay? So in essence, it deals with it that way. So that's what's happening with it here. In the brain, it's actually converted to methylhistamine, okay? And the histamine is working as a neurotransmitter, and you get essentially these products, okay, which is methylimidazole acetic acid, okay? All right? So,
just
yeah.
so you remember, in the lecture on regulated secretion, there was a whole slide showing histamine from mast cells that was released upon stimulus. There were nice electron micrographs of that. So if you want to go back and take a look, you can.
Okay. I didn't know that they had mast cells, but I
I
think...
use it as
What?
very pretty.
Yeah. They're pretty cells? Okay. I guess it must be getting time for the test. Okay. All right. Okay, so the last one I'm going to talk about is this strange neurotransmitter, which is nitrous oxide, nitric oxide, okay? Nitric oxide is made from, there's got to be a stimulator molecule or a nerve impulse, okay? So here's the neurotransmitter, and here's some stimulator substance. It's made from arginine and oxygen, and you get NO, okay? And it can be made from arginine and oxygen in this way, where it's stimulated. The enzyme is set up that way. You don't need to know it. And NO is then released, and it goes into smooth muscle cells in blood vessels, okay? And the NO stimulates guanylyl cyclase. And what does guanylyl cyclase do? Okay. They haven't done that. What does adenyl cyclase do? What does adenyl cyclase do? You're not making me happy about what I taught you in the, yeah? What? It makes CAM, right, it makes CAM. So what do you think guanylyl cyclase does? CGMP, right? So it's the CGMP, everybody see that, right? Okay. All right. So it can't be CAM. So we are going to get GTP goes to CGMP, which brings about relaxation of blood vessels. Okay. So when relaxations of blood vessels are going to the smooth muscle in that, what's going to happen to them? Are they going to contract or dilate? Dilate. Okay. Now, what were the two ways to increase cyclic AMP in a gluconeogenic liver cell? Somebody back there? Somebody over here spoke, and do you remember? Yeah. Okay, that would be the two different ways, but I was thinking in two different sites. You're at the receptor site, so that's good. Epinephrine or glucagon, okay? There was another way to do it. How about you coffee drinkers? Somebody said it already. She goes, did you say it? What happens? Why do people drink coffee in the morning? Because they want to increase their blood glucose levels, okay? So they increase their glass. So what in coffee? Coffee. What's the thing that you think is going to be the agent that's going to do something in coffee? Caffeine. Okay, and what did caffeine do? What's the general term? They're called methylxanthines. Guess what? They look like xanthine. Okay, do you remember we say xanthine? Okay, and what do they inhibit? Okay, let me ask you, how does cyclic AMP get broken down? By a thing. Good. That's the thing is an enzyme. Okay. It's something that ends in ACE. Okay. Anybody want to? Phosphodiesterase. Right. So guess what? There's a phosphodiesterase that breaks down cyclic GMP. Okay. Interestingly enough. Okay. All right. If it's in the correct area. Okay. There's an inhibitor. An inhibitor of, so what this is broken down to GMP, okay, by a phosphodiesterase, specific for cyclic GMP. There's a group of drugs that are inhibitors of this. Have you ever heard of Viagra? Have you ever heard of Cialis, with the two people sitting in the bathtubs, okay, looking out over the sunset? Okay. All right? What are they used for? Don't tell them it's my initials, okay? My initials are E and R. Okay. Why do people take the little blue pill? I know you can't imagine that you might need a pill for it, but okay. All right.
Oh, it's
For ER, erectile dysfunction, ED, and I'm sorry, thank you, it's ED, erectile dysfunction, okay? Okay. I got in trouble, by the way, for going farther on that in your school newspaper, your school newspaper, okay? All right. So, they're taken to, in essence, come on, just to go on, yeah. The reason for people taking it is that they want to dilate the blood vessels so that the hydrodynamic, right, will essentially function, okay? All right? And so that's really what this is, okay? And so you're actually interfering with it, okay? So it's a neurotransmitter. It also has a direct effect, okay? And so that's how Viagra and Cialis and those drugs work, okay? I know you can't think about it, you know, it's a problem. Neither can I.
Basically, it stimulates the creation of cyclic GMP, so you have relaxed blood.
Right, and so you're stimulating that, but what happens is I have a pool of this in the, all right, and now I have, this is normally broken down. If it can't be broken down, and I'm making it at the normal rate, the level of cyclic GMP goes up.
Also, you give them Viagra to break down the cGMP.
No, to inhibit the phosphodiesterase.
Oh, so the Viagra inhibits the phosphodiesterase.
And therefore,
You have an increase.
in cyclic GMP, you get further dilation. And then if it persists for 6 hours or more, it's a real problem. I think 2 weeks ago, I think in the Good Doctor, they had a guy who was.
I'm not caught up on that.
What?
I'm not
Caught up, okay. I think 2 weeks ago. What? Priapism, right. And the problem is that if it's for more than 6 hours, you can end up having necrosis occurring. Okay, and that's the side effect. I've gone too long, okay? So no, we're right on time, okay? So okay, so I'll be back. I'll be going.
Keep it down, guys. I have to say that I am no longer going to allow people to come in the middle of the second lecture or in the middle of class when we have a quiz. You have to be here in the room for the lecture because it's way too disruptive and it's really disrespectful to your lecturer with all the talking and the disruption that's going on. So that's it. You have to be here for the entire lecture.